Cytokines. Cytokines are especially important to the cell-mediated response, or the T cell response. They are chemical messengers that regulate other immune cell responses. For example, chemotaxis, natural killer activation, phagocyte activity. Some examples of some cytokines are interleukins. Interleukins are your communication between different leukocytes. So interleukin 1 stimulates the T helper cells in presence of antigens. Interleukin 2 activates the T and B cells. We've seen interferons involved in viral infections back in Chapter 9. Chemokines induce the migration of leukocytes. Cell-mediated immunity also develops from the stem cells. However, we know that T cells mature in the thymus. So they start out in the bone marrow, then they travel to the thymus where they mature, and then they migrate to the lymphatic system. T cells respond to intracellular antigens. So they respond to antigens by T cell receptors. TCRs are antigen receptors that are responsible for recognition and reaction. Remember, they are antigen specific, just like the B cells. The T cells basically differentiate into effector cells when they are stimulated by an antigen. So first, they recognize the antigen, and then you have something called thymic selection, which eliminates many immature T cells, and then those effector cells then differentiate into memory cells, the plasma cells. And of course, the plasma cells are the ones that are just the regular T cells. They don't differentiate into antibodies, because they don't make antibodies. 3 types of T cells: helper T cells, which are your CD4, cytotoxic T cells, which are your CD8, and they destroy target cells with perforin. And then you have regulatory T cells, which suppress other T cells. So let's start with the helper T cells first. A macrophage is also known as what we call an antigen-presenting cell, an APC. An antigen-presenting cell, such as a macrophage, comes in contact with an antigen and ingests it and processes it. So it eats it up and then it digests the antigen. From there, it displays the antigen on its surface with a self molecule to make MHC. So we talked about these major histocompatibility complex protein that sits on your surface of your cells. So the macrophage displays the antigen using this MHC. So the antigen fragments are on the APC surface with the MHC. The TCR on the T helper cell is going to recognize the antigen and the MHC2 on the antigen presenting cell. And when it recognizes it, it's going to bind to the MHC2 and stimulate the antigen presenting cell to produce interleukin 1, which is a cytokine. Interleukin 1 activates the T helper cell to make interleukin 2. Interleukin 2 then stimulates the maturation of the T helper cell. So it's going to go through and make more clones. Those mature T helper cell clones make other cytokines, which stimulates other T helper cells, cytotoxic T cells, and B cells. So let's put that together. So here's your antigen. That antigen is going to be eaten up by the antigen presenting cell, which is a macrophage. And it does so by phagocytosis, and we already went over the process of phagocytosis in Chapter 9. So it's going to take it in, and then it's going to digest it. And then it's going to display a piece of that antigen using the MHC molecule on its surface. So here's the MHC molecule and the antigen. And here's a T cell going by, T helper cell going by, and it's like, wait a second, I recognize this. And it recognizes it, it binds to it, and that causes the release of interleukin 1 and then the release of interleukin 2. And then that's how the cell goes ahead and continues to activate the immune response. The cytotoxic T cells destroy their target cells on contact. So the target cells are usually cells that are infected. Okay? So it's useful for destroying intracellular parasites that cannot be attacked by antibodies. Okay? So basically, the cell has been infected. So it's not on the outside, it's on the inside. Okay? And you know that B cells, the antibodies, can't get to the inside of a cell, so we need the T cells, preferably the cytotoxic T cell. So what the cytotoxic T cell does is it recognizes this MHC molecule class 1. So the infected cell is like, okay, I am sick. So what it's going to do, it's going to display this foreign antigen that has infected it on its surface using the self-MHC molecule. When it does this, the cytotoxic T cell is going to recognize and bind. Okay, it recognizes and binds to it, and then it's going to secrete that perforin. And that perforin is going to be secreted, and it's going to cause the cell to lyse. So hence you kill the infected cell. This picture is not in your book. It's one of my favorite pictures from how to describe that this is the picture from your book. It's not my favorite, but you can use it as well. So non-specific cellular components we have seen have become a part of cell-mediated immunity. We know that they're less specific than the T cells, but they help out. So we know the activated macrophages, right? They're activated by the antigen and by the T helper cells. So they're stimulated by ingesting the antigen or by cytokines. And then we also know about the natural killer cells. They also get involved, and they come in contact with the cell to lyse them. If we were to put the relationship of the cell-mediated and the humoral immunity together, when we talk about that T-dependent antigen, if it's T-dependent, it relies on the B cell. So sometimes the B cells need help from the T helper cells to make an antibody to an antigen. So before the B cell become activated, it needs permission from the T helper cell. And that's if we're dealing with the T-dependent antigens, which is majority of the time. And if we already talked about the antibody-dependent cell-mediated cytotoxicity, ADCC, when we were discussing humoral immunity. So what this picture shows is how everything ties together in this chapter. So even though we talked about humoral immunity by itself and cell-mediated immunity by itself, we'll see that the humoral and the cellular, the B cell, again, if it's a T-dependent antigen, needs permission from the T helper cell before it can even make a plasma cell-like antibody. And this is why acquiring HIV can be detrimental, because HIV takes out these T helper cells. Here ends Chapter 10.